Scientific communication helps in accurate and clear exchange of scientific knowledge. Therefore, this communication workshop aims at informing the participants about scientific methodology and polishing their communication skills. Let us welcome our guest speaker, Professor N.S. Punekar, who will be joining us at the workshop from IIT Bombay. Professor Punekar completed his PhD from Indian Institute of Science, Bangalore. He did his postdoctoral training at Institute for Enzyme Research, University of Wisconsin, USA. He has been working as a professor at IIT Bombay since 2001 and heads the Metabolism and Enzymology Lab. He also has worked as Dean of Academics and Students at IIT Dharwad. His area of interest include microbial biochemistry, molecular enzymology, microbial metabolic regulation, understanding the metabolism through biochemical and recombinant DNA techniques, fungal molecular genetics, and its application to metabolic engineering. He has also authored several books, for example, Enzyme Catalysis, Kinetics and Mechanisms, which has been highly cited. We are really excited to have with us Professor N.S. Punekar.
All right. Good morning, Professor Punekar. Can you hear me? Professor Punekar, you are muted.
Can you hear me?
Yes, I can hear you. Yeah, great.
So my video is also on. So I was just waiting. Okay, fine.
Great. Yeah. All right. So good morning, and thank you so much for accepting invitation to give talk today. Yeah. We are very excited to listen to your talk on research philosophy. And I'd like to mention to all the participants that I think maybe several years ago, maybe 4 or 5 years ago, I had opportunity to talk a course, jointly with Professor Punekar. We shared the course, and I was very much enthusiastic about even listening his lectures. That was my first time when I was talking about communication skill course, and it was really highly stimulating talk. So I thought to pass on that particular knowledge and that experience to all of you. And we are very glad that Professor Punekar has accepted, and he's here today to talk to all of you. So over to Professor Punekar.
Yeah, thank you. Good morning to all of you. And I'll see whether I can share the screen.
Yes.
That's important. Yeah.
On the top right side, that.
Yeah. Share. That's where I am. And I have something opened up already on the desktop. I'm just trying to work with it. Let me just see if I can. Can you see this now? No.
Not yet.
Not yet. Okay. All right. So I'll just probably do this other way around. Computer. Yes. Yeah. Okay. So here it goes. Okay, it's loading.
Okay.
Yeah, in the.
I think something is coming. Yes. Now we can see your slides. Great.
Okay. So I'm not very good at Teams. I mostly do with Google Meet. So this is the first time I'm doing.
Okay. No, it's working well. Yeah.
Can you see it? full screen? Can you see it, or it's okay?
It is on the full screen. It looks very good. Yeah.
Good. Okay. So again, for the audience, I will start by saying that go back to a history a little bit. In 1993-94, many of you may not have been born. Okay, we had a bright student, and he applied after his master's here to go to US. Okay, and he immediately got an offer. He joined a lab, and it was somewhere in not one of those big universities, but a smaller university, a small town university. He joined the lab, and in a year's time realized that it was worse than a lab in India, okay, in terms of research. So it was a very difficult transition for him, and he eventually had to leave that position and do something else. So the reason I am trying to bring this up is that, you need to be well prepared for doing research, and you need to be well versed in looking for what you really want, rather than just simply some, preconceived idea that research is this, research is that. Okay, and obviously there's a lot of preparation that goes in, even to make yourself think that you are suited for research or not in the first place. Because if research is a very long enterprise, at least 4 to 5 years of PhD, and at the last stage you realize you are not cut out for research, there are problems. Okay, so what I do is normally I talk to the students, particularly the fresh students who want to venture into research, something about research philosophy, and I do share that kind of a background with you, so hopefully it will be helpful. Okay, so let me begin by telling you that this particular research philosophy is a 45-50 minutes presentation, is a condensation of what I cover as a biological inquiry, history and philosophy course, or more recently it's called as research philosophy, BB 602 in IIT Bombay. Okay, part of that course. So these are the titles in which I try to cover, okay, to get the student to feel how and what of research, okay, and when and where of research first. So there are various titles here, as you can see, how to prepare, how to choose a problem, experimentation, chance, its role, hypothesis, ideas, imagination, intuition, reasoning, both inductive and deductive reasoning, observation, how do you make an observation, how you not to make an observation, and difficulties and strategy in doing research, and then obviously science and scientists, what's unique about them, and what's not so good or good about being a good scientist. Then obviously I talk about a little bit about research in biology, how it is different from research in other sciences. Okay, and obviously everything goes with ethics and biological research, which is very close to us because we are also biological species. So how you conduct your research, I'm sure some of you might be aware that there is a new bit of evidence coming up that the Wuhan virus was probably a part of a problem in doing research which came out of the lab, not probably out of wild. Okay, so we still don't know, but I think these are the kinds of things that should not happen or should not be encouraged. So obviously research ethics is an important issue. Okay, so I will not be able to cover all of it, but I thought I'll just give you a glimpse of what it means to be doing good research and what ingredients are involved in being a good researcher. Okay, so research preparation in the sense is a process of science. Okay, and in this process of science, it's important that the process is more important than the product. Okay, what I mean by that is, you should not be looking for a degree as an end objective itself. No, you should look at the process of how to do research is what you should learn, rather than just getting the degree at the end. So you get a degree, but you don't know what it is for, then obviously it's not worth it, right? Okay, so Claude Bernard, famous physiologist, pretty early in the 20th century, 30s, 1920s, he had this to say: those who do not know the torment of the unknown cannot have the joy of discovery. So in research, you need to be actually part of the discovery process, rather than just doing it as if it is a chore that has to be done, and it's not an exam. Okay, basically that's what's important to know. All right. So obviously you need a PhD degree for doing independent research. And obviously PhD degree is not very straightforward. As you can see that it involves a transformation, and obviously not this kind of a transformation, but you need to evolve yourself. Okay, it's a lot of hard work and effort. Okay, so but what is actually a PhD? This is Lee Shulman, President Emeritus of Carnegie Foundation. He had this to say about a PhD. A degree in morality, actually. Is it a degree in morality or degree in what? Okay, so we view the doctorate as a degree that exists at the junction of the intellectual and moral. So it's not just discovering something new, but discovering something new with particular moral and ethical background in it. Because you are always in a society and you do research in the context of the existing value system of the society. Okay, so junction of the intellectual and moral, the PhD recipient is expected to serve as a steward of his or her discipline or profession, dedicated to the integrity of its work in the generation, critique, transformation, transmission, and use of its knowledge. So it's a bit mouthful, but effectively this is how you define. You define when your doctor starts to practice, he has to take something called a Hippocratic oath. So probably this would be something which you need to be doing, you need to be aware of before you actually venture into doing research. Particularly, your qualification is a PhD to do independent research. Okay. Okay, so knowledge, actually understanding what do you do research about. Okay, knowledge is like a sphere. Okay, this is what Pascal has to say. The greater its volume, the larger its contact with the unknown. Okay, so this is just a nice picture to show you that in elementary school you seem to know almost everything. Then you go to high school, learn a little more. Then you get to bachelor's, master's. And eventually when you do a PhD, you just hit upon a very small area, very highly specialized, but very small area of research. And that small dent somewhere at the periphery is the PhD. And Bernal, the famous physicist, had this to say about philosophy and particularly research. The full area of ignorance is not mapped. We are at the present only exploring the fringes. Actually, a PhD is that, you need to get a nice background before you can. Okay, so I hope you can see this picture. Are you able to see this picture?
Yeah. It is clear.
So first of all, before you venture into doing research, you should be able to understand where the knowledge stands at that point of time. Okay, so where do you start from? What is it that we are? What do we start, and where do we start? Is what we have to begin with. Okay, so first step is to learn to understand literature and others' perspectives. So some people think research is this, some people think that the area is this much progressed, and what is it that we know about the area? Okay, and obviously the same research paper that when you read and when somebody else reads, something is wrong with this. Are you able to still see it, or? Hello?
Somehow got disconnected right now. It was visible.
I just got disconnected, so I'm just wondering what's.
Till the previous slide was okay. I think just now the screen is not seen.
Yeah, I'll probably load again. Okay.
Yeah. Please share it again.
Why it just went off by itself? Yeah, it's surprising.
Sometime, technical.
Yeah. In fact, I was wondering why. All right, so I'll load it again.
Yeah.
Okay. Does this happen often or first time?
Does not happen so often, but yeah, once in a while it can hang up.
Yeah, so I'm not sure. Anyway, I think it's getting loaded again. I'm not able to see it.
Yes, now it is coming. Yeah.
It's coming. Okay, it's a little slow, I think, to load.
Right. Yeah. No, it is fine. Yeah, it is on the full screen, but it is starting from the first slide, so you will have to skip.
No, I'll move it. Okay. And also getting this pop-up on my screen all the time about people joining. So I don't know how to.
Actually, our team is admitting them. That is a little bit, disturbing.
No, yeah, I know, I'm able to see it all the time. So, okay. So whenever you read a research paper and particularly prepare yourself for your own area, so you see it in a particular way, right? And different people see the same piece of data, the same piece of research in different ways. So to understand literature is an important first step. So I show a picture here. I'm sure many of you see something, right? What do you see here? Since it is not interactive session, so I might as well tell you that it looks like an African landscape with a hut and some trees and some people sitting here. Okay, but if you just, this is one perspective of this picture, and if you move it 90 degrees, this is what you see, the same picture. Okay, so basically, this is a surrealistic picture by Salvador Dali, and it's called Paranoid Head. So if you look at this way, it looks like head, but if you look like this, it would look like a landscape. Okay. So the point of it is that different people gather different bits of information from the same piece of research. Okay. So it depends on your taste, your interest, and how you take it from there. So that's something which is important. So I think the good thing is to be able to understand most of the perspectives, if not all, before you start, and so that you know where the cutting edge of that particular field is. Okay, and it also requires an aptitude to be able to do good research. Okay, and this is something which I'm very particular that the researcher should know before they start. So what do you mean by aptitude? So here are some questions, and here are some answers to these questions. And this is in Chinese, obviously. How many of you know this for this question? This is the answer. There are two ways of learning this. I mean, the two ways of knowing this. One thing is to be able to know what this actually means in Chinese and what this actually means in Chinese. But the other quicker way of doing it is simply memorize this picture, memorize this picture. For this picture, this is the answer, because this picture is a question. So this is one way of doing it. But this is not the way research should be done, can be done, should ever be done. Okay. So in fact, you need to understand before you do something. Okay. So I don't know why it's moving. Okay. Yeah, so here it is. Actually, this means moon, this means sun. So there are relationships, south, north, fire, water. Unless you know this information, you'll be just a picture. So you'll be by rote learning, you are doing science. So that's not what should be done. Okay, in research, you need to really understand the system before you can get more answers. Okay, or even contribute to research. This is extremely important. And that's why originality in research is extremely important. And I'll give you this example of Parkinson and Thompson, two students, okay, from the old British Times, English Times. I don't know how many of you know about Parkinson, how many of you know about Thompson. You may have read them in the textbooks, but you may not remember them by this name. But I'll tell you this small story. Okay, so this was in those days, there was a school, there was a good teacher in the school, and both of them were good students in the class. Okay, and the teacher was teacher's favorite was Parkinson. He always read a lot of things. He did a lot of extra reading, and he was very meticulous. So he always topped the examination. Okay, Thompson, on the other hand, was not so meticulous, but he was also pretty good in what he was doing, and he was, in terms of his own contribution, he was original in his answers and things like that. So just to be cut the story short, the teacher put up a question paper in the final exam with about 10 marks extra questions where it was outside the syllabus. Okay, and he wanted to really make sure that he caught the best student in the class. Okay, so it turns out, as usual, Parkinson got the highest marks, and he also answered the extra question, whereas Thompson also came out with a brilliant answer for the same question. Okay, and the teacher now got worried, concerned that how come Thompson can answer this question because it's outside the syllabus, but he understood, and he was biased that Parkinson was a good student, so he understood. So he called up Thompson and said, look, this was an outside syllabus question, and you were able to answer it. So I know Parkinson answered it because he reads a lot, he reads many things. How come you did this? Okay, how were you able to do? Did you copy it? Is what he asked Thompson. Thomson had to say only one thing. No, that particular piece of paper from where you asked this question was written by me. He actually wrote that paper. So that is the kind of originality that Thomson had. You know who Thomson is? Lord Kelvin. I'm sure many of you know the Kelvin, temperature scale. The Lord Kelvin, so Thomson. So the point of it is that Parkinson was very good at rote learning and exams, but Thomson was very good at research, discovery, and originality. Okay, so I think in research, to research as a career, I think one has to be more like Thomson. It's important to be Parkinson to some extent, but I think one has to have a lot of original research ideas. Okay, and preparation. This is extremely important. Originality of research is extremely important to be successful in research. Okay. The second ingredient for being successful in research is you should always find a good mentor, because research is something which research cannot be done by yourself. Research is more like an apprenticeship. That means you go to somebody who knows how to do research, and this is you start right from the beginning. Michelangelo, Da Vinci, you name people right from the beginning, Newton. So people always had a mentor from where they learned. So it's important to realize that you should find a good mentor. How to choose. Somebody asked a question, how to apply. Okay, it's one thing to apply, but it's another thing to be in the right place. Okay, with the right mentor. Okay, and this is a very important step before you actually embark on doing good research. So the best teachers are those who show you where to look, but don't tell you what to see. Okay, so they won't bias you, but they allow you to grow intellectually. Okay, that's extremely important. So you should be able to find a good mentor, and this comes with a lot of your own inputs as a person. Okay, so I'll just show you a couple of examples of that. And here is Alexander von Humboldt. I'm sure many of you understood Humboldt Fellowship. Okay, from Germany. So he was a natural scientist, and he worked with George Christoph Lichtenberg, who was also his mentor. Okay, and Humboldt had this to say about his own mentor: I do not merely regard the sum of positive insights that I was able to gather from what you told me. He's talking about Lichtenberg. Okay. What I value even more is the general direction that my train of thoughts took under your guidance. Okay, truth in itself is precious, but even more precious is the skill to find it. So you need a good mentor to be able to train yourself like this. It's extremely important. Okay, it's not just enough to be fashionable. Join a lab which is successful, or join a lab which is doing some very fancy, fashionable research, or join a group where there's a lot of publications coming out. It's important to be able to join a lab where you can learn. Okay, and different people learn differently. So you need to find out what is a good mentor for you rather than just going by fashions. Okay, so this is something important. Time memorial, tested. Okay, so here is a picture I have synthesized just to show you that a good mentor creates a good school. Good mentor produces intellectual progeny, which are very powerful in terms of success. Okay, so here is Adolf von Baer, and this is Baer family in terms. This is a German school. Von Bayer's strain theory, some of you must have heard in chemistry. Okay, so from his school, he had 4 great students, Wistater, Wieland, Fischer, and Buchner. Okay, Buchner is the same person, Buchner, who talked about fermentation. All right, and then these 4 had their own students. Like Wistater had Kuhn, Wieland had Fridolin, Emil Fischer. The person who did a lot of work with carbohydrates, sugars. Okay, he had these students: Diels Adler, from him, Warburg, Otto Warburg, respiration, remember, glycolysis, Windus and Buchanan, and Warburg students were Meyerhoff, okay, Krebs, Theorel, Hans Krebs, PCA cycle, Theorel, redox enzymes, Meyerhoff, EMP, Emden, Meyerhoff, Parnas pathways, so basically glycolysis, Lippmann, fatty acid metabolism, Ochoa, again, nucleotide metabolism. So you can see that what is common to all of them is that they come from one great school, and they have learned from some real great teacher, and it has been percolated down over generations. This is an intellectual family, they are not blood relations, but they're intellectual relatives in terms of father, child, and like that, parent and progeny. There are people who are chemists, strictly chemists. Okay, so here you have all those names in black are chemists. All those in red were people who were both chemists and biologists. Okay, and then you can see in obviously blue color code, they're all biochemists and later on biologists. So what is common for all of them? From this school, every one of them is a Nobel laureate. And I think you learn a lot by being in a place, being with somebody who is an excellent mentor. Okay, that's what I'm trying to point out. It's extremely important for you. Okay, so this is again what I call it as the passage effect, that if you go to a good lab, good group, and learn under a good teacher, you will be a good researcher. This is extremely important. Okay, so you may have a lab where there are a lot of facilities, techniques, but if the mentor is not good, or you may have a very good mentor, but the lab is not very well equipped, either ways you are difficulties. But I think a mentor is more important than the facilities, because mentor will always find ways, or he will let you have ways of doing research, even under limitations. Okay, so a good mentor. Here is an example of Carl and Gertie Corey. Okay, I'm sure many of you know about Corey cycle. Okay, the pyruvate and the lactate turnover from muscles to the liver and back. Okay, so these glycogen metabolism and Corey cycle, Carl and Gertie Corey, and the kind of people who went through their labs in Cincinnati, in US, Arthur Kornberg, Severo Ochoa, Sutherland, Leloy, Christian Didouet, and Edwin Krebs. So each one of them have done different kinds of biological research, but what is common to all of them is they spent substantial amount of their time honing their skills of research under these headship, or under these headship. Okay, so the good mentors, good researchers will impart knowledge, and it just you imbibe very good value system for doing research. So this is an important ingredient in research, obviously. All right. So I call it a passage effect. So you need to have spent some time in a good lab, good group, good mentor. Tutelage of a good mentor, I think will do much better. Okay. And this is tested time again. Again, I repeat that this is because research is more of a apprenticeship. It cannot be done in a classroom setting. So if you are interested in research, you need to be giving good attention to this kind of a requirement for good growth of your in your career. All right. So another aspect of is that you should have good imagination, originality, good mentors, but there has to be some limits as well in science, because science is not literature, science is not poetry, science is not something else, but science has some limits, and this is set out by our own thought process, because we want to be logical about it. Let the imagination go, guiding it by judgment and principle, but holding it in and directing it by experiment. So this is what Michael Faraday had to say, this is where the art differs from science. Art and science are both intellectually exciting, inspiring things for human endeavor, but in science, you actually direct it through experiment. You have limits. If it's not true, you cannot. Your imagination has to be quelled, and your imagination has to explain nature rather than create nature. In some sense. All right. So this comes time and again. You have experiments that almost always have a preference for the outcome of an experimental. Experimenters almost always have a preference for an outcome. That means I can think that this experiment should work like this, but in reality, it may not happen, because nature has its own ways and there are own rules and sets, answers honestly. Okay, so it's important to understand that you cannot fool nature, but you have to understand nature, and that's the difference between art and science, right? Okay, so slaying of a beautiful hypothesis by an ugly fact. So you may have a great hypothesis, but if there are no data to support it, then obviously you have to ditch the hypothesis, not the data, right? And you can remember that Frederick Waller did urea synthesis, okay? And by this particular simple experiment, vital force theory was simply tossed out, and it was a very powerful theory at that time. Okay, Waller's synthesis of urea by ammonium cyanide really made sure that there is nothing like vital force. Biological systems also behave and are under the guidance of physics and chemistry. Okay, so it's important to understand. The obviously you need to do experiments. You need to have controls. You need to know when the experiment is successfully conducted and the results are of value and can be trusted. Okay, and this is important. And there's a need for controls. This research philosophy obviously works on controls. In fact, I used to hear from my mentor that in research, 95% of the time it is the controls. Only 5 to 10% of the time it's the actual experiment, test versus control. So 95% of the time you do controls to make sure that the 5% of the time or 10% of the time what you do is actually properly understood in the context. Okay, and that is the reality. So there is a need for controls, in good controls in doing biological research, any research for that. So in controlled experiment, what do? We take test versus control. We do randomization within versus between experiments. We do pilot versus siting experiments versus dry runs. You know, these are all important points for doing good research, because biological research is extremely expensive, okay, in today's context. And if you design an experiment, do an experiment, and then realize it was wrongly done, I think a lot of time and money is wasted. So that's why you need to do a dry run. You need to do a pilot experiment. You need to plan experiments before you actually execute them. Okay, extremely important. Okay. So you cannot have sloppy and ill-planned experiments. But at the same time, I would say that by chance, if you do that, and if you still end up doing a failed experiment, sometimes they are also useful if you analyze them very well, carefully. If you analyze them carefully, they're very useful. Okay. Obviously, the data, when it is large number of data points, data sets, statistics, biometrics come important. Obviously, everything else is dictated by common sense. That's important. In research, we do what is called as method of reduction. Okay. So we design experiments in such a way that there are many things that can vary, but we vary one thing at a time. If many things are varied at the same time, then you don't know why the outcome is. Is it because of one or the other parameter that they have changed? So in the reduction, the method of reduction is important for science. And that means you vary one thing at a time and figure out what happens as a function of time, function of temperature, function of this, function of concentration, whatever it is. So you do what is called as follow the method of reduction. That means one thing at a time is changed. You design experiments in such a way that you get a very clear-cut answer, and it can be cause and consequence. Cause and effect relationship can be established. So that method of reduction is important. Reduction as a method is important for science. Reduction as a philosophy may or may not be important, particularly in biology. So I will not elaborate on this, but just to tell you that what happens at the molecular level need not always be the explanation for a gross biological outcome. Okay, so there are birds, for example, there are fish, for example, who go in a shoal, or birds fly in a group. There is a population of animals, insects, which behave in a particular way. You will not always find a gene for these things. Okay, sometimes the effect is due to multiple genes. It's what is called as a emerging property in biological systems. And method of reduction will work to do good experiments, but reduction as a philosophy does not work very well in biology. So you need to work at different levels. So method reduction is a good tool for research, but reduction as a philosophy is not a great help in understanding and doing research in biology. We could spend more time on this later, but that's something which I want to mention, particularly with respect to controlled experiments. Okay, so here is an example of a controlled experiment. Obviously, some of you must have seen such pictures. Okay, so this is the guy, Dr. Raj. The device, you say, will ensure glitch-free launch of, and here he has this, chilies and lemon device to make sure that this obviously does a successful launch. Science and faith are not the same. Obviously, you need to be sure that you design your experiment so that it can work very well. Okay, and it can give you a result which is clear-cut, and there is no controversy, there is no uncertainty in that result. So clean results, what one looks for. The controls are important. So in fact, the scientific method, evolution of an experimental design, has gone through a lot of changes over centuries. Okay, so I'm not going to read all this up, but just to show you that it has evolved over a lot of effort by human endeavor. You can see that there are something about library, then you have the falsification experiments, repeatability of the experiment. So this is a very heavy slide. I will let you actually think of and imagine that how actually the method itself has progressed. And obviously then you have double-blind experiments, placebos. In today's context, I think a lot of experimental design goes into good experimental design goes into clean experiment outcome. So start from collection of data, library, Alexandria. You have people who showed that replicability is important. Okay. The whole range of evolution has taken place in doing good research. And so the method itself has how to do science itself has evolved over time. Okay, so here are a couple of quotations related to that. No experiment is a complete failure. It can always serve as a bad example. And obviously Thomas Edison, I have not failed. I have just found 10,000 ways that won't work, but eventually the lamp worked. Incandescent lamp worked. Okay, so you have to observe and analyze failures. Okay, and quite often opportunities that you get, even after very good design of experiments, okay, are disguised results. Some you have to work very hard to decipher them. So most people don't recognize them, and they just dump an experiment because they are not able to understand. So it's important to spend a little bit of time to be able to observe and analyze, even if they are failed experiments. A lot of experiments. I'll show you some examples where people contributed a lot to be and thought a lot, and they contributed a lot to science by doing some very incisive thinking, okay, and observation. So I'll show you some examples of that. Okay, so obviously, if Fleming had simply thrown the moldy plate, we would not have had penicillin today, right? So he was able to use even something which was meant for discarding, and he was able to apply his mind and get something out of it. Okay, so I'm sure you are familiar with the story of penicillin, okay, and Alexander Fleming. All right. So this is another way how you should not do experiments, or you should not design experiments. Leo Szilard is again a famous physicist. So he would say, if you do stupid experiments and finish one a year, some experiments take long time, obviously. You finish one a year, it can take 50 years, let's say. Okay, but if you stop doing experiments for a little while and think how it works, how should it be designed, and how quickly it can reach there are only about 5 different ways of doing it, and not 50 different ways. And it will take only a few experiments to distinguish them and quickly arrive at the result. Okay, that means thinking, designing clean experiments, thinking is a very important component of that. So that's what I think people highlight, particularly when you're doing biological research. Okay, and that's why I think somebody said aptly, look before you leap, right? Similarly, think before you clone, because today anybody can clone anything, but is it worth cloning? Is it required? And is it useful? Does it give you an unambiguous answer? That's very important for us. Okay, so whatever is said by Leo Szilard applies very well to biology today. Okay, so make best use of your chance is what I said. No, whenever, even if an experiment has failed or something, your experiment has not given you the result which you anticipated, still it can be of value. So whatever chance you get, you should make best use of it. So first of all, there are infrequent opportunities to observe something. So increase this by increasing the number of times you do experiments, different kinds. You have to notice the clues to be alert, interpret the clues, prepared mind. You should have a prepared mind. You have understood something. So if you are prepared in physics, it's very unlikely that you can contribute to cutting edge in biology, unless that particular biological problem requires that physical tool. Okay, so this is something which you'll appreciate when you see how a prepared mind is required. Okay. Obviously, Alexander Fleming was looking for microbiological interactions when he saw that Petri plate which has to be discarded. So the prepared mind. I'll give you a couple of more examples you'll appreciate. Okay, distinguish between promising ones from dead ends and false leaves. So a famous scientist sometimes said that you should quickly realize that you are not sitting on a boiled egg. It's not going to hatch. You cannot incubate a boiled egg and expect it to hatch, right? So you should know when to stop pushing the wall. Okay, so you should also know how to regroup. And these are very important, ingredients in being a good researcher. When you do get a good chance, when you do get a good observation, exploit it thoroughly. And if you can't exploit it, then obviously somebody else will exploit it. See, we all associate penicillin with Fleming, right? But Fleming discovered only the phenomenon of antibiosis, and he showed that penicillin exists. But it took almost 30, 40 years because he could not follow it up. He could, 30, 40. It turns out that in 2000, the first decade of 2000, 2010 up to 2010, then the Australian scientists discovered that Helicobacter pylori was responsible for peptic ulcers. Okay, so Kornberg actually had the discovery almost 50 years ahead of the time. Okay, but you can't help it sometimes. You see, this is what happens in research. But we have to be aware of these things, that these things do happen, and you have to make best use of chance that you have. Okay, that's something very important. Okay, so here are some examples I want to show you. Okay, tryptophan was discovered by Golland Hopkins by chance. Bacterial staining was discovered by chance by Christian Graham. I'm sure you know Graham staining. Diabetes and the role of pancreas were discovered by Minkowski, again by chance. Lacteals, or the fat digestion, were discovered by Christian Bernard, again by chance. Protein crystals were first got by Kornberg, Arthur Kornberg, when he was an enzymologist before he worked on DNA polymerase. So that was by chance. Bacteria near the edge led to the discovery of the phenomenon that there are aerobic and anaerobic organisms by Louis Pasteur. So this whole list, at least limited list, tells you that you can also make your own list like this. All these discoveries were chance discoveries, but the people had prepared mind and incisive thinking process, so they were able to take it to completion. I mean, look at Pasteur. He simply thought that there were bacteria running away from the edge of the droplet of the under the microscope, and he thought that maybe they are running away from oxygen, and he figured out fermentation. He figured out anaerobic bacteria and things like that. It's amazing. So one thing in research, important for research, is history. History of how things happen is more important than actual material. Okay, and history is a great teacher, and I would encourage students who would like to do research to find out about how things came about, how people discovered something, rather than actually just memorizing the facts. Facts are important, but in today's world, you can download all the facts and you can go to any fact by Google. But you need to know how it came about, and that tells you the process of research, process of discovery, and that's very important for us. And that is some kind of an apparent mentorship, that you will get. It's extremely important to know, how things happened historically. All right. Okay. So great scientists are always excellent observers. I already showed you the examples in the last slide. Here you have an example of Claude Bernard. And of course, example of Louis Pasteur, okay, soil, earthworms, and then he eventually figured out that anthrax was coming through these kind of a cycle, and Pasteur's discovery of butyric acid, fermentation, and anaerobic bacteria. And obviously in physics, you can see Röntgen discovered X-rays by chance, movement of the needle, electromagnetism by Faraday. Okay, so you see a lot of these things. I'll spend a little bit of time on the first example. You see how observations are critical, okay, and how these things contribute to good research. And you have to be a good observer. You cannot be sloppy, and you cannot be careless. You have to have full attention. Then only you can become a researcher. In fact, it's said that if you are going to work with a problem, you should internalize that research problem so that you are thinking it all the time, including in your sleep, so to speak. Okay. So here is an example of Claude Bernard. What did he do? He was working with rabbits, and he used to get rabbits for his work from a particular vendor. And this vendor would give him the rabbits. He would keep them for some time, and then eventually he would experiment with them. And he was observing the urine of these rabbits as a routine and for some purpose. And he figured out that normally rabbits excrete what is called the turbid and alkaline urine. Okay, turbid urine. They're herbivorous animals. Okay, so one bunch of rabbits suddenly he got were actually not doing this. They were excreting clear and acid urine. Okay, this is more like carnivorous animals. people, animals which eat meat, okay, which hunt and eat meat, okay. So this particular process of just looking at the urine set a train of thoughts in his mind, and he was an excellent observer. He figured out why is it happening. So why is a rabbit behaving like a, let's say, a cat in some sense, okay. So he quickly went back and figured out this happened because the rabbits were starved for almost a week. For some reason, the supplier had starved them this time and not fed them. And since they were starved, the rabbits were starved, so they were using internal protein material, so they were actually digesting their own internal material. So there was emancipation of these rabbits, and they were mobilizing their own protein material, and that their urine looked like carnivore. So eventually he did an experiment where he fed the rabbits with protein diet, meat diet, and not force them, and then they behaved like carnivores. The urine was again clear, and so based on all this, what was he able to discover? He was able to discover the whole process of digestion. So he discovered bile, bile fluids, where exactly it comes out into the digestive system. So one observation of urine, whether the urine of the rabbit was clear or alkaline, he was able to go through the whole discovery. Claude Bernard, you can Google up and find out how many discoveries he made in physiology, particularly animal physiology, which eventually led to human physiology. Chyle, digestion of fat, role of bile, pancreas, a lot of this contributed by Claude Bernard. And I think the starting point was just observing a rabbit which was not behaving normally in terms of its urine output. Okay, so it's chance observation, but how you can exploit that's extremely important. Okay. Yeah, and obviously when you do good research, you need to publish. I'm sure writing skills are also going to be discovered as a part of this workshop. But it's important to know that you should publish your work, because people need to know what you have done, and people don't have to reinvent the wheel, particularly if something has been done. So I would read this Charles Darwin's quotation, why publishing is so important. Observing the intricate functioning of nature is sheer pleasure. So doing research is sheer pleasure. For people who love research, it's sheer pleasure. It's an ecstasy. Okay, writing is sheer pain. Obviously, it's very difficult to write. Sometimes you have writer's block. Don't know where to start, how to write. But if you don't set down on paper what we have observed, okay, how are we going to document the miracle of evolution? This is what Darwin said. So unless you set it down on paper, you will not be able to tell others that, look, this is how nature works. No, this is what I have discovered. It's extremely important to be able to do good communication. You should be able to do good writing. Okay, good writing. It's extremely important to communicate, both written and verbal, because unless you can tell people that you have done something, it's not possible that your science will flourish. Your research will actually get accepted, appreciated. Okay, so it's extremely important to do publishing, proper publications. Okay. So this is an example again. I want to tell you about a young composer once came to Rossini with the score of an opera and asked him to place a cross at each mistake that he could find. Okay, so Rossini was a famous composer. Okay, Western classical music. When the young man returned several weeks later and saw no crosses on the manuscript that he had given to Rossini, he asked very, very happily, "Oh, then there are no mistakes in my score." Then Rossini said, "My dear friend, if I had made a cross at each mistake, this would not be an opera. It would be a cemetery. So there will be so many crosses that they will look like a cemetery." So you understand that writing is extremely important, and writing well is extremely important. Okay, and obviously many of you, if you end up getting into doing good research, you will write annual progress seminar reports, you will write your manuscripts, you will write your thesis. See, it is important to not cut paste, but it is important to write lucidly, succinctly, crisply. Okay. In fact, I remember my own mentor once told me, okay, I use the word short-lived. Okay, for a phenomenon I was writing up. I said this is short-lived. Like mRNAs are short-lived. Okay, so it was replaced by a word called ephemeral. Okay, so it's a good, nice-looking word. It's more fashionable word, but more importantly, that my mentor told me that short-lived has one letter extra than ephemeral. I hope you understand what I'm saying. So even when you write, there are a lot of considerations. People go to such details as to make it succinct, make it short, make it beautiful. Okay, so it's important to communicate your research well. Okay, and many good researchers. I don't know how many of you have heard about Richard Feynman. Feynman lectures, excellent lectures. So he was a great communicator, good writer, and good speaker. So you should read one of his books. Surely you are joking, by Mr. Richard Feynman. It tells you how very short sentences people are able to communicate very well. Okay, extremely important for doing good research. So unless you communicate, people don't know that you have done research, right? You have contributed to research. It's important. So I'll quickly go through a few aspects of science and scientists, what it means to be a scientist. Okay. So this is how science progresses. So I just want to show you this process of science in this very simple slide. A complicated hypothesis, which usually entails an element of mystery and several unnecessary assumptions, come out first. Okay. Normally a complicated hypothesis comes. This is opposed by a more simple explanation, which contains no unnecessary assumptions. This is how it evolves. And the complicated one is always the popular one first, but a simpler one as a rule, okay, eventually is found to be correct. This process frequently requires many years, actually many years. So always a complicated hypothesis eventually gets simplified, and minimum kind of a description is required to show that it's beautiful. You can see that all Newton's laws of physics can be very simple, one simple equation at a time. Okay. Describes the whole phenomenon. Okay, so that is how things work. Again, it blocked. I don't know why it blocked. Are you able to see this screen?
Just not dropped again.
Again, it dropped. It's dropped again. I'll go back. Go back. I have to upload it again. This is surprising. What has happened today?
Yeah, today the teams look like today.
Something is wrong. I think I've done this 4th time now, but I'll try it again. Let me see.
Sorry for so much trouble in between.
No issues. Actually, no, I'm only concerned about the discontinuity for the audience.
That's right, yeah.
Okay, let's see if I can. Because if this drops and they are not dropped off, it's fine. Only the slides have dropped off.
Right. Only
then after they start loading, and they're waiting in the lobby, quite a few of them. So
yeah, I think team is allowing them. Yeah. Now we can see the slide is coming.
Yeah. Okay. Let's see again. I'm loading it. I have to go forward. So with all this, how much time do I have? I have to also probably.
No, please take your time and finish it off. But there is no rush.
Okay. So then that's what I mean. I didn't want to.
There was a 15 minutes break as well, which we can also use.
I'll probably. There are 44 slides, but I think I'll cut it short.
No, please finish it off. I think they would love to listen to your talk.
Yeah. Okay. So normally, that is how science progresses. So people have a lot of bits of information. They don't understand it, so they make a nice big story. But eventually, the story becomes very crisp and very pleasing, aesthetically very pleasing. Okay, that's how science progresses. In fact, Max Planck had this to say about the process of this science progress. Okay, scientists never change their minds, but they eventually die. Okay, that means that scientists have some favorite hypothesis, favorite picture about what the phenomenon is or what that particular topic is. But then, they new and new evidence comes in. You have to improve the quality of the picture. Okay, and then get the most recent picture of what that phenomenon is all about. Okay, so understanding science, understanding a phenomenon is more like an asymptote. You keep adding to it. More new evidence comes in, you have to improve your hypothesis to accommodate all that. Okay, so that's something very important that happens all the time. And but obviously, a favorite hypothesis, somebody stuck to it. Very difficult for that person to come out of it. And that split personality has to exist in every scientist. That you have a favorite, but at the same time, you should know that it is my favorite, but may not be true all the time. Okay, additional evidence, you have to change the hypothesis. So hypothesis testing is a very important concept in science research. And obviously, you have to have an open mind that new evidence comes in, you should be willing to change or drop the hypothesis but not drop the data. Okay, that's something very important. Okay, so there are a large number of scientists today on this earth. Research has become a profession. Okay, so more than half the scientists who ever lived during the entire history of the world are alive today. So a lot of people are getting into research. Okay, it used to be a hobby, and today it has become a profession. Okay, it used to be a hobby. Okay, so once there are so many people, getting into the act of doing research, then there will be a rat race. Okay, and the rat race. The problem with the rat race is that even if you win, you are still a rat. Okay, so obviously, a rat race is not something which is very conducive for good research. Healthy research. So this is something which you have to appreciate. Okay, outcome is important, but outcome is important in the context of a healthy activity. Okay, because there are otherwise a lot of pressures. So, what kind of scientists are there? Although there are so many scientists, what kind of scientists are there? We can actually group them into 4 different kinds, at least on the face of it. The first stage is a new concept of ideas come, in science, and you can think of Mendeleev and the periodic table as an example, the DNA structure of Watson and Crick as an example. So new concepts, totally new ideas. So scientists contribute to this. In the second stage, you build techniques and language of that science, particularly that area, like Sanger, 2 sequencing tools to understand proteins, understand nucleic acids. Lamely, for example, some of you might know electrophoresis. Okay, then Altschul, the internet and the comparison of protein sequences. So the bioinformatics that was contributed by. So in the second stage, usually techniques and language of science is built. In the third stage of scientists with new tools, insights, more questions, and most of the time people work in this area. Scientists very rarely do they get new concepts and ideas, because you keep looking at good, excellent quality science, you build new tools, ask more questions. Some of them lead to something novel. Okay, but most of these are average scientists. Okay, so they contribute, bit by bit, in delta quantities they add to the knowledge. And there is a 4th stage of scientists, okay, where you look at the literature. The literature has grown very rapidly. You organize that literature, summarize it, and review it, and make it accessible to a fresh and budding scientist. See, for example, suppose if you did not have a textbook, okay, where would you start your research? Suppose you did not have a good review of a field, where would you start your research? So obviously it's more challenging, right? So the 4th stage of the type of scientist actually is where people organize the whole literature. I remember when I was a student, the book by Yvonne Wright, Immunology, was just about 250 pages, 300 pages book. Today, the same immunology book is probably 1,000 pages or more than 1,000 pages, maybe 2-3 volumes. So obviously, there is an explosion of knowledge. And when there is so much data that comes in because of a lot of research done by the 3rd stage, you will have to summarize it, make it acceptable, make it understandable for people. And obviously, you need to organize, summarize, and review the entire literature. And 2 examples I can give you is Benjamin Levin, Genes, the book on genes, right? Excellent book. Today, I think it's 12th to 13th edition is out there. Okay, and then Peter Atkins, Physical Chemistry, again, an excellent book. So all the principles are brought together, and you synthesize the field into a whole. Okay, so that is the understanding. So I put these 4 levels, but please make sure that this is not the hierarchy. This is just the 4 levels of organization. So you need all kinds of people, and now you can see where you can contribute to as a researcher, as a scientist. It is not necessary that everybody can contribute here, will contribute here. Some of the people who are contributing here end up coming here because they do a lot of basic, fundamental research, new tools, insights, more questions are asked, but some of them become very interesting. So they actually move here. You also need people like Benjamin Levin and Atkins, so people who can understand everybody else's work, synthesize it, make it presentable. Okay, so this is the kind of scientists you can get. So it takes all kinds to progress. Okay, so scientists actually are in some sense hunters. What do they hunt? They hunt microbes, they hunt vitamins, they hunt enzymes. Eventually, today they hunt genes. Okay, so this is the progress of biology. In fact, in some sense, you have seen. Okay, and I would suggest that to look at the process of science and how scientists actually worked I would recommend that you read something, a book on microbes, a book by Paul de Kroof called Microbe Hunters. Excellent book. I think it's a very good read to get you a historical perspective of how microbiology, particularly the infectious diseases science, has evolved. Okay, so it's important. Okay, so I would. So what are the incentives for us to do research? Why should we be doing research? See, it is one thing that, look, I went to school, I went to a college, then okay, after bachelor's, I have to do a master's, a PhD. So it just goes on. Is that what one is doing, or one is genuinely interested? Okay, so the incentive to do research and do like when one likes research for different reasons. And I just listed those kinds of reasons here. So the basic reason why somebody wants to research, the urge to satisfy curiosity. Okay, so there is a creative instinct. One wants to create, do a beautiful painting. One wants to do nice poetry. One wants to write a nice novel. But in science, also there is curiosity. There is also creativity. So you want to create something, understand something new in nature. That is a second instinct that you have. Which incentive that you have to do good research. Okay, and desire to create new knowledge, better understanding of nature. Okay, and obviously there is some applied aspect to it in some sense that you want to do something good. Your understanding, your research help betterment of people, accomplish good for the mankind. A lot of people come, okay, I want to cure cancer, I want to find a solution for this particular disease, I want to grow more food in a small area of land, things like that. So a lot of and then obviously there are genuinely humans who would like to get recognition. They want to get become famous. They want to get counted as somebody who is a great scientist, so gain recognition. So obviously there is also making money is an aspect of it, because today there is also some applied research which will allow you to make money by actually patenting the invention. Okay, so these are the driving forces for doing research by a researcher. Obviously, what is in the name, why people want to be famous. So this is an example. Okay, so here's an example of a bug. Okay, in fact, this bug, let me see if I can. Okay, this bug was discovered by, I'll just tell you the name of the person, is probably in the next slide. Okay, Garrison. Okay, this is a bug. Actually, I consider this an extreme honor. This is what he has to say. He discovered this bug and he says this. Besides, I knew no one was going to write and ask to name a few species of swan after my name. Beautiful swan, but nobody will name. So I am happy with my name associated with this particular bug. You have to grab these opportunities when they come along. So Stragophyllus gallisoni is this bug, and the Garrison discovered this, so his name goes with that particular bug. Okay, so naming is something which people like. They get, I'm sure you can see here a big list. I'm not going to read through it. Theodore Escherichia, Escherichia coli, that Albert Ludwig Neisser, Neisseria gonorrhea, you have many examples, Donald, Daniel Elmer Salmon, Salmonella typhimurium, so typhoid fever, gastroenteritis. So the point is that many of these people incidentally got their name immortalized on these organisms. Okay, similarly, you have Planck's constant, Planck's name comes in, Avogadro number, Dalton, molecular weight is now given in Dalton's, Ohm's law, Ohm, okay, Newton, the force, okay. So actually, obviously, one becomes famous by associating their names with a discovery. I'm sure all of you know LB broth, okay, Luria Bertini broth. So this broth is discovery of Luria Bertini, so it's called LB broth. So the name obviously is an important incentive for scientists. Okay, so recognition and awards are important, but I think they are overplayed in today's context. Okay, so one has to be very careful. In fact, this is where the problem lies today, because the competition is so bad. I said about rat race. Eventually you are a rat if you always take participation in such a rat race. Okay, so a little bit about research and ethics. Okay, so every one of the researchers generates data, okay, through experimentation, okay, some or the other kind of data, and interprets it, okay, for a living in some sense. Okay, so here is a list of things that you can do as a researcher. You can generate data which is of sloppy quality, meaningless data. you have generated data but do not know what its meaning is, you can become a technician. You don't care what it is, but you generate good data and give it to somebody. Okay, or you generate good data, but you're a researcher, but you misinterpret it. If you misinterpret it on purpose, then you are cheating. You are misinterpreting because of personal bias, or you can be an honest mistake. So any of these are possible. Okay, you can analyze it right, and you are a decent researcher. Okay, if you do generate good data and analyze it right, you are a decent researcher. But you analyze it right, and also with a further insight, and probably give a nice idea, which turns out to be true later. Then it opens up a new area. Then you are a genius. Okay, so that's how it works in research. All right. Okay, so obviously, you cannot fix, and you cannot cheat. Okay, so honest mistake is acceptable, but cheating is not. You have a lot of examples of that in literature. Okay, you have to be careful. Okay, so Samuel Johnson, famous, politician and statesman, had this to say: integrity without knowledge is weak and useless, and knowledge without integrity is dangerous and deadly. So I think important part of research is also integrity. Okay, you have to do good research, but do research with ethics, extremely important. Okay, and obviously we should not confuse science with technology. Acceptance of technology depends on ethical and ethnic value systems. So obviously, you know that today there are vaccines. You have a choice of vaccines for COVID. Now, the question is, you cannot force somebody to take only one vaccine. When there is a choice, you should let it go. Okay, and let people decide what kind of vaccine they want, as an example. Okay, so it depends on the ethnic and ethical value systems. So if you're in China, you are forced to take a particular vaccine. But most of the 3 countries, you don't have to, you can have a choice. So unfortunately, what has happened over time is that our scientific power has outgrown our spiritual power. We have guided missiles and misguided men. So Martin Luther King has to say this about, scientific ethics, okay, integrity and scientific ethics. Obviously, you must have heard of the story of the lion makers from Panchatantra. I'll not go into details, just to tell you that you need specialists to assemble the bone of the lion, to put meat on it, body on it, and also eventually put life into it. But what is more important is that you also need a person who will be able to tell you that really is it worth doing it or getting killed by the live lion. Okay, so the story of Panchatantra, I'm sure. So a bit of common sense is very important in being a good researcher. All right. So obviously, research is a collaborative venture. Okay, it requires teamwork. You need many people to contribute. There was a time when there was a single author doing publications, doing research, and then publishing it. Today, you see that there are a lot of authors on a publication simply because there is so much to be done to get one particular idea, confirmed or proved today. Okay. So you will find that. Okay, research is a collaborative venture. So this is a paper in Nature 2007, very old, but already the concept was there, that the demise of the lone author by Matt Green. Okay, what he had to say was this, that this is a paper from Nature Genetics. You see the number of authors in this? The number of authors is covering almost a full page and 3 or 4 different organizations. Okay, genome-wide association studies and things like that. Okay, so contribution by these many authors. If I were to pick up any one of the author from here, I want to know what he or she did. It's going to be extremely difficult. Okay, it has its issues, with what is the contribution of the person. So should you actually get a name on the paper? Have you contributed sufficiently to get a name on the paper? It's a very, again, important ethical issue for publications. Okay, so a researcher cannot be just a pair of hands to do someone else's bidding. Intellectual inputs distinguish a technician from a true researcher, and we should know exactly whether we deserve to be on the paper. Okay, and there are something like, I don't have a slide to show you, but there are a concept of what's called as child authors coming up in China. In a sense that some of these big scientists and big politicians, they have their children, okay, whose name is put on publications. They're not even in college. They're put on publications because by the time they come to college, their CV looks good. They have not done anything just because their parents put the name on some of the publications. So they have a list of publications to show by the time they go for admission. Okay. And again, in China, if you want to practice medicine after MBBS, you need to have 2 publications. Okay. So people are pushing to get publications by hook or crook. So obviously that destroys the quality of research, quality of science. So I would end up by telling you, having seen all that I covered so far, it's a platter. Why it's a platter? Because there are so many aspects to research. The research philosophy involves many things: how to prepare, where to go, what to do, how not to do things, how to do things, how to observe, and then how to publish, how not to publish, how to take credit, how not to take credit. So there are a lot of issues related to research. Okay, and it is an apprenticeship, as I said. So I want to leave you with the impression and the question that, are you cut out for research? You should ask this question before you venture in. Just because somebody else is doing research because it's fashionable, you should not go into doing research. Okay, the philosophy of research is such that only if it is your calling and if you really enjoy doing it. All right. So I'll stop here. If you think some questions can be answered or discussion, I can take. Otherwise, we'll stop here.
I think we can ask the participant to raise their hands if they have any questions or comments.
Yeah.
Yeah.
I'm still sorry that there were so many breaks because it also disturbed my flow in some sense.
Right. But I think still all the ideas were well conveyed. So thank you so much, Punekar.
Yeah.
Let's Saichiran, you want to ask a question? Then Vaishnavi after that.
Yeah, sure.
Saichiran, go ahead. Unmute yourself.
Thank you for the talk again, sir. It's been a couple of years since I took your course, and it felt very nice to revisit the slides again, the lecture again.
Yeah.
Sir, I have this query. I just wanted to know your opinion on why results that are deemed negative or not on expected lines, why is such work not published very often? Like, what is your take as a researcher as to why such work usually doesn't come online? It would actually help many researchers avoid experiments which have probably not worked out in the past.
This is again a philosophical question at one level, and it is also a question that scientists are also human beings. Okay, and that is something which you should appreciate. That people want credit.
Yes.
Okay, people want credit, and people want to claim more than what they have actually done quite often. And unfortunately, this is a human nature. Okay, they want to show that they have done something so great that everybody should appreciate that. All right. In the process of doing this, there is a competition. And the competition is that I have done better than somebody else. Okay. So in the process, what goes to the bottom of the pile is that negative data. Even if it is excellent quality negative data, it goes to the bottom of the pile.
Right.
Yeah. So if you look at the discussion, there is something called DORA, Declaration of Research Assessment. Okay, in California, research assessment. So you can Google up and find out. It's called DORA, Declaration of Research Assessment. That should we really use impact factors to evaluate somebody's contribution or not? Is a question. A researcher, should we use? How do we evaluate good research from not so good research? Okay. So I think impact factors came into being some time ago, and people are finding it very hard that impact factors can actually contribute. Anything in terms of doing, a good assessment. Extremely difficult. Because there are some fields of science where the best journal in the field, the impact factor is 1 or less than 1. But there are some, like in biology today, there are some journals whose impact factor is probably obnoxiously high, 40 plus, 50 plus, 60 plus. So how do you compare these two kinds of research?
Exactly.
Okay. So sometimes the research is fashionable, sometimes it is not. Okay. And just because the research is not fashionable does not mean it is not important. Okay. And who is to blame for all this? The research community, right? Exactly. It's not the layman who understands it. It's us who understand this problem. We need to sort it from inside. Okay. And one other aspect of this, which you didn't ask, but I'll tell you, is that how do we evaluate a research output? You do what is called as a review of a manuscript, right? Okay. So what is the norm today? The norm today is it is a single blind. It's a single blind, and there are editorial boards and editors, and there are also reviewers, and this is a captive audience, and there is a turf defined by every group. Okay, so they define, and if you want to be part of that club, you get in, otherwise you don't get published very easily. And it's a fact of life. Everybody knows it. Okay, so people may claim I got this publication here, I may got publication there, but the fact of life is that it is also a reflection on the club, which club you belong to, or where you are, where you come from. Okay, what kind of associations you have. So ideally, I think people are moving in this direction. Ideally, you should have a double blind review. Not a single blind review. Ideally, you should have a double blind review. Okay, what do you mean by a double blind review? So the manuscript should go out there. You should not know who has written it. I mean, somebody should moderate it, of course. It cannot be so off the logic. But a manuscript is submitted without the author being explicitly displayed, and a manuscript is evaluated by somebody whom the author does not know. This match has to be made. Then there is no bias. Today, even if the manuscript is, submitted by an author, okay, the author does not know who the reviewer is. There could be a bias. Okay, and I'm also telling you that I have reviewed some manuscripts where even without the author's name is given, I can figure out by the kind of, what do you call, literature citations they do, and the kind of work they do, I know where it is coming from. Okay, it's a big challenge. Understand what I'm saying? So publishing negative data, okay, is one step even behind what I'm talking about right now.
So perhaps we should not use the term negative data. Data is still data. I mean, it probably doesn't fit a particular answer question, but it's still data, right?
Yeah, it is. So there are challenges like this even. Okay. So I think it has to come out of this, and it will come out only when scientists don't make tall claims. They make make rational, realistic claims rather than making tall claims. See, there is money involved at the end of it, funding involved at the end of it. So there is always somebody who will go and make a big claim. Okay, so they probably will get money. And the moment a scientist, I mean, this is without any particular person in mind, the moment a scientist sells himself or herself to a politician for funding, everything is gone. Because you can claim that I will do this for you, and then you can't do it, or you do it but it's not acceptable scientifically, then everybody's in trouble. Our credibility is at stake. So this happens. This happens quite often. This happens. In fact, if you look at the Wuhan story now that is playing out in the public domain, that NIH funded some of that research. Okay, and there are people who wrote letters in support of that research, and it was actually propaganda. But now we are getting to know that it was propaganda. You see what I'm saying?
Yes.
So,
yes, sir.
Hundreds of thousands of dollars spent on research, which backfires.
Yes.
So I think unless one is clear about that, it's extremely difficult. See, this is one thing where science and faith have things in common. That is, in faith you have to follow, in science you have to follow. Okay. In faith, if you don't follow, you become a heretic. You are out of that faith. Okay. In science, if you don't follow, you're not easily thrown out. Unfortunately, you still are there, hanging in there somehow.
Right. No, great. Thank you so much, Mr. Punekar, for sharing some of the very practical points, and we have a lot of experience in this area. And just for the participants, I think some of the journals have started now the double-blinded peer review process, as Mr. Punekar mentioned. Now they're also asking you to remove the the name and identifiers. and only sent to editor, and remaining part can only be sent to the reviewer. That is actually a good start, because otherwise, lot of times there is a bias in the review process as well.
True.
I think we can see a lot of hands raised, but Professor Punekar, due to the shortage of time, we have now slightly running late of the session now. So Professor Devjani is waiting, and next speaker is also there. So we will stop this session. The participants, please write your questions in case Professor Punekar is able to reply back over there, or I can ask him on the email. We'll get back to you on some of your queries. I think you have a lot of intriguing questions and things for him, but sorry, we have to stop it now because we are running late on the session. So Punekar, thank you so much for giving this wonderful lecture on research philosophy and giving everybody some very practical tips of it. I'm sure they're all going to immensely benefit from your lecture. Thank you so much.
Just one last bit. If you can share my email ID with them, I have no problem. If they have a question, I can answer them.
Well, that's a wonderful thing, which can have. So we will definitely share with them your email ID, and
there may be questions, so I don't mind answering them offline later.
Absolutely, they're all very happy. See, like so much hands clapping for you. Thank you, Professor Punekar. Thank you so much.
Thanks.
All right, great.
Right. Good luck to you guys.